Before we move on to our last topic, which is reproduction, I thought it would be useful to review meiosis and some basic heredity or genetics. I say review because I'm hoping that much of this is material you've seen before. Sex determination. So we did talk about this a little bit before when we talked about the endocrine system. You have 23 distinct chromosomes, and you got one set of 23 from your father and one set of 23 from your mother. You have 22 pairs of autosomes. These are the body chromosomes, they're sometimes called. And then you have one pair of sex chromosomes. If you're male, you have an X and a Y for your sex chromosomes. If you're female, you have two X's. Now there can be variations of that. It is possible to have an extra sex chromosome, but that's a case of aneuploidy. Aneuploidy is where you have an unusual or abnormal number of chromosomes. So what you're seeing in this photo here at the bottom is the X chromosome and the Y chromosome. Notice that the Y chromosome is much smaller. The X chromosome has about 1,000 genes on it. They're all quite important. The Y chromosome has only a few genes on it. The most important one is the SRY gene, and it is the gene that tells the gonads to develop into testes, and then that brings about the production of testosterone, and that brings about the changes that we associate with maleness. Note on the karyotype here, remember karyotype is just a photo that displays all the chromosomes seen at metaphase in one of your cells. On this karyotype, we have 22 autosomes, and then here we have the sex chromosome. So this individual would be a male. So karyotype, I think we did talk about this earlier. A karyotype is prepared by taking a cell that is in metaphase and then dyeing it so that we can see the different parts of the chromosome, and then lining up the chromosomes and making this map, essentially. And we can use this to look for abnormalities. So for instance, chromosome 21, if we had another copy of that here, then that would indicate Down syndrome, also known as trisomy 21. So what you do is you take your metaphase cell and then you dye the chromosomes with a stain that will recognize areas that have a lot of C and G bases. And you can use fluorescent dyes for this, as we've done here, or you can just use a monochromatic dye that will just give you a black and white image. Keep in mind that when we're looking at metaphase chromosomes, they're going to consist of two parts. They're going to consist of two chromatids, and we've paired up chromosomes here that each consist of two chromatids. So sex determination is pretty straightforward in mammals like ourselves. There's actually lots of different ways to determine sex, but we won't get into how it works in the rest of the plant and animal and fungi system. But in mammals like ourselves, if there's two X's, that means that the embryo will develop as female. If there's an X and a Y, that means that the embryo will develop as male. So it's going to be the sperm that will ultimately determine the sex of the zygote and the individual that it develops into. So the eggs are haploid, of course, and they will have one X chromosome. The sperm, half of them will have an X and half of them will have a Y. From conception until about 6 weeks of development, the embryo is neither male or female. It hasn't differentiated yet. It contains what are said to be bipotential tissues. Bi meaning two, so these tissues have the potential to become ovaries and female structures, or testes and male structures. Whether or not the embryo develops as a female or male is going to depend on whether or not the Y chromosome is absent or present. So if there's a Y chromosome, The Y chromosome contains a specific gene known as the SRY gene, and it codes for a protein known as the SRY protein. And this protein is responsible for bringing about the development of testes, and then those testes will release testosterone, which brings about a whole bunch of other changes. Incidentally, SRY stands for sex determining region on the Y chromosome, kind of a clunky acronym. Now, another thing to note is that in this bipotential embryo, we have two series of ducts. We have what are called the Wolffian ducts, and they develop into the male-specific aspects of the sexual plumbing, I guess you could say. We also have Mullerian ducts, and they develop into the female-specific parts of the reproductive plumbing. Within the early embryo, we have a structure known as the urogenital ridge. At the 6-week stage, this urogenital ridge contains a bipotential gonad. So the gonad at this stage has the potential to become an ovary or a testes. Even at this stage, there are 3 distinct sets of cells. So we have a population of cells that will give rise to support cells in the mature gonad. We also have what are called primordial germ cells. Now, unfortunately, the word germ has a few different meanings in biology. We're not talking about bacteria or pathogens here. A germ cell is a gamete or a cell that will give rise to gametes through meiosis. And then we also have a population of steroidogenic precursors. These are cells that will give rise to cells that are endocrine in nature and produce steroid hormones. So looking at the differentiated gonads, in the female, in the ovary, we have follicular cells. Those are our support cells. They will support developing gametes, but they also produce the hormones estrogen and progesterone. In the male, these support cells are known as Sertoli cells. They help support the developing sperm cells, and they also release a hormone that's known as MIS. And we'll come back to that in a moment. The primordial germ cells are going to give rise to oocytes in the ovary, and these are cells that will undergo meiosis to produce egg cells. In the male, they're going to give rise to prospermatogonia, and these are cells that will undergo meiosis ultimately to produce sperm cells. The steroidogenic precursors are going to give rise to theca cells in the female, and they produce a very small amount of androgen. An androgen is a hormone that brings about male characteristics. Andros means male in Greek. So even in females, we have a small amount of these hormones that are rather important for development of body mass and so on. That's something we'll talk about again later. And then finally, these steroidogenic precursors in males are going to produce Leydig cells, and they are responsible for producing testosterone. And it's testosterone that brings about the major changes that distinguish between the two sexes. I have to admit, I've crammed a lot of information onto this one slide. Let's take a bit of time and break it down. And we'll start with the diagram on the left. Now, if you're female, you have 2 X chromosomes. You got one from your mother and one from your father. If you're male, you have 1 X chromosome that you got from your mother and 1 Y chromosome that you got from your father. The Y chromosome doesn't have a whole lot of information on it. It's got a few genes. One really important one, and that is the SRY or TDF gene. The X chromosome, on the other hand, has over 1,000 genes. But that SRY gene on the Y chromosome is responsible for bringing about male characteristics. It's also known as the TDF gene, which stands for the testes determining factor, because it brings about the creation of the testes. If the SRY or TDF gene is not present, the embryo will develop as a female. That's the default pathway. But if that gene is present, the ovaries will develop into testes instead. The testes will produce testosterone, and that brings about other changes. Okay, so that SRY or TDF gene is going to serve as a master gene. It actually codes for a protein that's a transcription factor, and that protein will turn off a whole bunch of genes, the ovarian genes, the genes that are needed to generate ovaries, and it will turn on a whole bunch of other genes that are involved in making testicles. So all of these genes are present, whether you're male or female, just they're turned on or off based on whether or not that SRY gene is present. The SRY gene is also going to turn on another master gene known as SOX9. Don't worry about that at all. Just realize that SOX9 is another gene that codes for a transcription factor, and it's going to turn on more testicular genes and turn off more ovarian genes. So if this one gene, SRY, is present in the very early embryo, it is going to impact the activities of about 4,000 other genes, which is pretty amazing. So that one gene will have a big impact. Okay, let's look at the diagram at the top. So within the area that's going to give rise to the reproductive organs, we have what's known as the genital ridge. Within this area, if there's no Y chromosome, the gonad that's forming there, the bipotential gonad, will develop into an ovary. If there is a Y chromosome, that Y chromosome is going to have this SRY or TDF gene, and that is going to tell that bipotential gonad to develop into a testes. So we'll have testicles developing. They're going to have two populations of cells, Sertoli cells that will produce something called Mullerian inhibiting substance, MIS, and we'll come back to that, and also Leydig cells that produce testosterone that brings about further changes. We also have germ cells. If it's an ovary, those germ cells are going to develop into egg cells, ova, and if it's a testicle, then these germ cells are going to give rise ultimately to sperm cells. Now, one thing that's really quite interesting that I'll get to in just a second is that males and females have the same parts. We really do. The parts are just arranged a bit differently. When we develop as very early embryos, we also have the same plumbing. We have the same ducts. So we have these sex ducts. And we have two sets of ducts as early embryos. And it doesn't matter if you're going to develop into a male or a female. You have these two systems in place. There are Mullerian ducts, and the Mullerian ducts will ultimately give rise to the uterus, the fallopian tubes or oviducts, and the upper portion of the vagina. Now, if you are male, those ducts will degenerate. There are also Wolffian ducts, and they give rise to the epididymis, the vas deferens, the seminal vesicles, the ejaculatory duct. They will persist if you're male, but they will degenerate if you're female. So the diagram at the bottom there, the bottom right, shows these two ducts. We have these Mullerian ducts, which are here, and also here. They're going to persist in the female, and you can see down the bottom that they're going to give rise to the uterine tube or fallopian tube, the uterus, and the upper part of the vagina. Now in males, we have these Wolffian ducts that are shown in green. They are going to persist. They are going to break down in females. In males, they're going to give rise to the ductus deferens or vas deferens that you see here. So again, we start with the same pieces, but they develop a bit differently. In the male, in the early testes, we have the secretion of something called MIS, Mullerian inhibiting substance, and that's what causes the Mullerian ducts to break down in males. Let's also look at what's happening externally. We'll look at the development of the external genitalia. And as I mentioned, males and females start out with the same bits. At the top in the middle, you're seeing the bipotential stage. This is a 6-week-old fetus. It's neither male or female at this point. At this point in development, the fetus actually has a very pronounced tail, and that's being cut off down the bottom here in case you're wondering what that stump is. The tail has been sectioned through so that we can have a view of what's going on. Above that, we have the genitalia. We have the genital tubercle. Tubercle means a nub or projection. That's going to develop into the head or glands of the penis or into the clitoris. So those parts are analogous. They are the same. They develop in a similar way. Underneath that, we have the urethral groove. So we have this little cleft that is going to develop into the cleft between the labia and the opening of the vagina in females. And in males, it's going to seal over and it's going to give rise to this ridge here along the scrotum and the underside of the penis known as the raphe. We also have swellings on either side of the urethral groove. These swellings will develop into the labia in females, they develop into the scrotum in males, and then of course the gonads are going to drop down into the scrotum in males. They're going to stay put in females. So in both males and females, we start out with one opening, the cloaca, and that's going to separate out into the anus and then also into the urethral groove. And then we're going to get the development of the labia and the clitoris in females. In males, we have the same structures. The only real difference is that the glans is going to move to the tip of a shaft and the urethra is going to migrate into the glans. But otherwise, we have the same structures. They're just arranged a little differently. Take a look at the women in this photograph and try to figure out what they have in common. You'll probably be surprised to learn that they all have a Y chromosome. Genetically speaking, they're male. But of course, they're not male. What's happened here is that each one of these women has a functional Y chromosome with a functional SRY gene. However, they lack functional testosterone receptors. So the gene that codes for the testosterone receptor is not functional. So what that means is during early development, they produced the SRY protein that caused the gonads to develop into testes. The testes produced testosterone, but there was no response. None of the cells that had receptors could respond. The default pathway for the embryo is female. So in the absence of testosterone or in the absence of a response to testosterone, the embryo will develop as female. Now it's also worth noting that the testicles themselves have testosterone receptors. Again, those weren't working in these individuals. There's a positive feedback loop that occurs. So if testosterone is present, the testes continue to develop and eventually drop down into the scrotum. Without testosterone present, the testes degenerated. This condition is known as androgen insensitivity syndrome, or AIS. Andros in Greek means manly. An androgen is a hormone that brings about male characteristics. These individuals are insensitive to those hormones because they lack functional receptors for them. Somewhat ironically, the mutation that causes this occurs on the X chromosome. That's where we find the gene for the testosterone receptor. Now remember that these individuals do develop testes, and during early development, those testes are producing testosterone, even though it's having no effect, and they're also producing MIS, Mullerian inhibitory substance. That is having an effect. That will shut down the generation of the uterus, the fallopian tubes, or oviducts, and the upper portion of the vagina. So these individuals, although in all appearances they are female, they are sterile because they lack those inner components of the female reproductive system. The woman on the right here, Eden Atwood, is an individual that has AIS, and she's rather outspoken about it. She's also quite a talented jazz singer, and you can find videos of her singing and also talking about this condition on YouTube. On to part 2, the production of gametes. So gametes are produced by a special type of cell division called meiosis. Meiosis only occurs in the gonads, and the gonads, of course, are the ovaries or testes. The ovaries and testes produce gametes, and they also produce hormones. And we're not going to talk too much about the hormones just yet. We'll come back to that when we talk about the reproductive system and its anatomy. So I'm hoping at this point that you are familiar with mitosis and meiosis. If not, make sure you take some time to review them, because you can expect to see some questions about this on the exam. So what we've got here is a hypothetical animal that has diploid cells with 6 chromosomes. So that top middle cell is the parent cell. It could be a body cell, or it could be a germline cell, which is the cells that are found in the gonads that give rise to gametes. But this is a hypothetical cell with the condition 2n equals 6. The 2n tells us it's diploid. 6 is the number of chromosomes. And note that we have 2 sets of homologous chromosomes. So there are 2 long chromosomes. One is red, one is blue. Those are homologous chromosomes. They contain the same information. We have 2 sets of short chromosomes. One is red, one is blue. Those are homologous. And then we have 2 sets of medium-sized chromosomes. One's red, one's blue. Again, those are homologous chromosomes. They contain the same information. Let's just assume that we got the blue set from our father and the red set from our mother. So again, we got 2 full sets of information, one from our mother, one from our father. Now, let's imagine we're dealing with the skin here, and this cell is going to undergo mitosis. What's going to happen is it's going to pass through the S phase of interphase. That's the DNA synthesis phase. And those chromosomes are going to be replicated. So after this happens, all of the chromosomes consist of 2 parts. Each chromosome consists of 2 chromatids. Now, as we enter into metaphase, these chromosomes are going to line up end to end along the metaphase plate, the middle of the cell. We have the spindle, which consists of microtubules that will attach to the chromatids, and then the chromatids will walk along the spindle until they get to either pole. So the chromatids should separate from each other, and at the end of this division, we should end up with 2 cells that are identical to our starting parent cell. Okay, now let's take a look at meiosis. So let's say that our parent cell in this case is one of the germline cells that's found in the testes, for instance. The cell is going to undergo the S phase of interphase, and we're going to have those chromosomes being replicated, so they consist of 2 chromatids. Remember, a chromatid is a single piece of DNA that's wrapped around a bunch of protein. Now, during prophase of meiosis 1, something unusual happens. The homologous chromosomes, the chromosomes that contain the same information, come together. And we end up with these clumps of chromosomes that are known as either bivalents or tetrads. You can use either term. So tetrad means 4. So we've got 4 homologous chromatids all paired up. And we can have crossing over between non-sister chromatids. So the 2 chromatids that make up one homologous chromosome are known as sister chromatids. But we can have crossing over, the swapping of bits of homologous information between non-sister chromatids. At metaphase, we have these tetrads that are lined up along the metaphase plate. So notice we're not dealing with individual chromosomes end to end. Now we have tetrads of homologous chromosomes along the metaphase plate. They're going to be pulled apart. So during the first division of meiosis, we have the chromosomes being pulled apart. And then we have a second division. And in the second division, the chromatids are going to be pulled apart. So the second division of meiosis is a lot like mitosis, except that we're starting out with half as many chromosomes because we've already reduced the amount of material. Finally, when meiosis is done, we're going to end up with haploid cells. They only have 1 set of information. And notice that we have swapped bits and pieces of information between the two sets that we started with. So when you were formed as a zygote, you had one complete set of information from your mother and one complete set from your father. And the information was on separate chromosomes. But when you make your own gametes, you actually take the information you got from your parents and you reshuffle it. So now on some chromosomes, you will have a mixture of your father's and mother's information. So all of the gametes are a little bit different. And that's one of the things that causes the diversity that we see in human and other populations. It's estimated that two humans mating to produce offspring can produce a diversity of individuals that's actually greater than the diversity of humans seen on the planet. So just the statistics, how many different outcomes we can get, it's quite amazing. Now keep in mind, this is a hypothetical example. We're only using 6 chromosomes because it's easier. In humans, that parent cell would have the condition 2n equals 46. The function of meiosis is to have the ploidy. So we're going to cut the ploidy in half. The ploidy is the number of sets of chromosomes that you have. So our bodies are diploid and multicellular. Our gametes are haploid and single-celled. Fertilization is going to unite two gametes, a sperm and an egg, and that creates a zygote, which is diploid. So we've reinstated the diploid condition in the next generation, and that zygote will divide by mitosis to produce a multicellular diploid organism. Let's do a quick overview of gametogenesis. Gametogenesis is the process by which gametes are made. Remember that meiosis consists of 2 divisions, meiosis 1 and 2, and we start with 1 diploid cell and we get 4 haploid cells. Or at least that's what we've said so far. Things are a bit different in females. But let's start with spermatogenesis, that's the production of sperm in males. We have what's called the primary spermatocyte. That's a diploid cell, so that's the parent cell, and it is part of what's called the germline of cells. Those are the cells that ultimately give rise to gametes, and those are cells that are maintained in the testes. So these primary spermatocytes are only going to exist in the testes. The first division of meiosis will produce 2 haploid cells, and those cells are known as secondary spermatocytes. The second division of meiosis will produce 4 haploid cells, and those cells are known as spermatids, and they will develop into the mature sperm. Okay, let's look at the female side of things. This process is called oogenesis. Oo means egg. Whenever you see 2 O's stuck together like that at the beginning of a word, it's generally something to do with eggs. We have our primary oocyte, and it is diploid, and it would, of course, develop within the gonad, and it develops actually quite early during development. It develops in the embryo quite a long time prior to birth, but we'll come back to that in a bit. What's going to happen is this cell is going to complete the first round of meiosis, so meiosis 1. That's going to happen every month. Every month you're going to have 1 egg that completes meiosis. Now it divides into 2 cells, 2 haploid cells, but notice that the division is asymmetric. One cell contains almost all of the cytoplasm. The other cell got very little of it. The cell with almost no cytoplasm is called the first polar body, and it's not going to contribute to the production of eggs, basically. It's going to abort, as we'll see. The cell that got all the cytoplasm is known as the secondary oocyte. This is the cell that's going to be released into the fallopian tube or oviduct, and it's going to travel down the oviduct, and if sperm is present, we have fertilization occurring. When this fertilization occurs, that prompts this secondary oocyte to undergo the second round of meiosis. So meiosis 2 will occur, and it is also asymmetric. So again, we have one cell that's going to get all the cytoplasm. That is the cell that the sperm is actually attached to. And then we're going to have a another cell that receives very little cytoplasm, and it becomes our second polar body. So at this stage, we have the sperm fused with this large egg cell, which is the ovum, and we have these two polar bodies with very little cytoplasm stuck to the side of the egg. The nucleus of the sperm cell is going to fuse with the nucleus of the ovum, and that's going to give rise to a diploid zygote. The polar bodies are just going to disintegrate. Let's take a closer look at the genetic material. You know that it's DNA, but chromosomes are not just DNA. In fact, they're half DNA and half protein. And the protein serves as kind of a scaffold that the DNA can be wound around. DNA is a rather fragile molecule, and you have about 2 meters of it packed into every cell in your body, which is incredible because your cells are much, much smaller than 2 meters. So that DNA has to be condensed, and it's condensed by wrapping it around proteins. What you're seeing in this image here is a cell that's been stained with a red dye that will bind to the cytoskeleton, and then a blue dye that will bind to the chromosomes. The material that makes up the chromosomes, so that mixture of DNA and protein, is known as chromatin. And we won't get into this in a ton of detail, but basically you've got DNA, and the DNA is wrapped around proteins known as histones. 8 of these proteins stick together to form a ball, and then the DNA is wrapped around those balls. What you're seeing in the picture here, this is taken with an electron microscope, and you're seeing a naked strand of DNA. DNA doesn't usually occur like that in your body. Instead, DNA is wrapped around these balls, and that's what you're seeing here. So each one of those balls contains 8 histones, and DNA is wrapped around it about 2.5 times. And then there's a bit of naked DNA in between these nucleosomes. So if we look at that under the electron microscope, we see what's sometimes called a string of pearls, because it looks like a bunch of pearls strung together. Active DNA, DNA that's being decoded and being used, looks like this. As the enzymes that go along, like DNA polymerase and RNA polymerase, as they go along and they read that information or copy it, they just unwind a few of these nucleotides at a time. So we have these 8 core histones, and then we have this other histone called histone H1, which will clamp the DNA to that nucleosome. Now, why is this important? Well, DNA has to be condensed remarkably prior to cell division, and what happens is all those histones come together. So the DNA is wrapped around those histones. The histones are positively charged. DNA has a slightly negative charge, so they stick together quite nicely. What happens next is that H1 histone, special histone, comes along and clamps the DNA to that octamer, that ball of 8 histones. And then the histone H1s are attracted to each other, and they wind these balls of protein and DNA into a very tight spiral. The image on the right shows you what this looks like top down. So the histone H1s are going to form these nucleosomes into a spiral structure. So here we have what's called a solenoid, and I'm not going to have you remember the name of that, but these are all the balls of histone and DNA twisted into a spiral. And the next thing that happens is that spiral of DNA and protein is going to be attached to scaffold proteins and thrown into short little loops. And then that structure is also going to be looped again. So the end result is that the DNA is condensed remarkably to 1/10,000th of its original length. And this all has to happen during the preparation for division. So just another view of that. Again, there's more detail here than you need to know. I do want you to know what a histone is. I want you to know that that's used to package DNA. And I want you to know that the DNA condenses prior to cell division. So again, we can see the solenoid, that is our DNA wrapped around proteins, and then the proteins packed together. And then we can see that this solenoid is attached to this red scaffold structure, which is also made of protein. And then that is spiraled up as well. So it's quite a complex process that has to occur. When people first started studying chromosomes, they realized that they were 50% protein and 50% DNA. And for quite a long time, people thought it was the protein that was responsible for coding genetic information, and that the DNA was actually there as a scaffold to help condense the protein. But now, of course, we know it's the other way around. So a chromosome is a mixture of DNA and protein, and the protein is there to compact the chromosome so that it will fit into the nucleus and so that the chromosomes can be aligned on the metaphase plate and not get all tangled up, because again, DNA is a very long, very fragile molecule. Now, the important thing to note is that if we have a chromosome that contains just one piece of DNA, we call that a chromosome, but if we have a chromosome that consists of two pieces of DNA, we still call that one chromosome. In that case, we have one chromosome that contains two chromatids, and that's what you're seeing in the second diagram here. So at the top here, this is a chromosome, but this is also a chromosome beneath it. After the S phase of interphase, the chromosomes in your cells will consist of two sister chromatids. Each chromatid is one piece of highly condensed DNA, and they're attached at what's called a centromere. So it's a little narrowing, generally in the middle of the chromosome, but it can be towards one of the ends. The next thing that's going to occur, of course, is that those chromosomes are going to be pulled apart. And if we're just talking about mitosis, this is a one-step process, and we end up with our two daughter chromosomes. The term sister chromatid refers to chromatids that are in the same chromosome. If we've got, let's say, a tetrad, it's going to have two chromosomes. Non-sister chromatids are chromatids that are not in the same chromosome. So chromosomes can occur singly or as a pair of sister chromatids. Sister chromatids are joined at the centromere, and they separate during mitosis. Within the centromere, we have something called the kinetochore. It contains motor proteins that will grab on to the mitotic spindle and pull the chromosomes towards the ends of the cell. The mitotic spindle is actually kind of like a set of railway tracks. The chromosomes actually run along it, so they're walked along it. It's not like a system of ropes. It's not actually a tug of war that's occurring. Some more terminology. So ploidy, as you should know by now, is the number of sets of homologous chromosomes in a cell. Now, one thing that's important to note is that, let's say we've got a cell that's got 6 chromosomes, and we've got 2 sets of 3. Now, let's say that this cell enters into S phase, and now after that, it's still going to have 6 chromosomes, but each one of those is going to be composed of 2 chromatids. The number of chromosomes has not changed, and that means the ploidy has not changed, even though the amount of DNA in the cell has doubled. So the ploidy refers to the number of sets of chromosomes, and those chromosomes can be composed of a single piece of DNA or 2 pieces of DNA. So if you notice in that earlier slide where we were talking about gametogenesis, after the first division of meiosis, we did actually have the ploidy. That is the division that cuts the number of chromosomes in half, but the chromosomes are still made up of 2 pieces of DNA. The second division of meiosis is going to pull apart the chromatids, and we haven't changed the number of chromosomes, but now each of the chromosomes consists of 1 piece of DNA. C value refers to the amount of DNA. You can have 2 different ploidies in human cells. They can be haploid or diploid. We can have 4 different C values. The lowest C value you can have is in a gamete. So in a final egg cell or a final spermatozoan cell. They have a C value of 1. If we've got a cell that is diploid, but not undergoing division, it is going to have a value of 2. So a cell in G1 of interphase is going to have a C value of 2. A cell that has gone through the S phase So it's replicated its DNA. So all of its chromosomes consist of 2 pieces, has a C value of 4. So again, a gamete, C value of 1, a body cell that's in G1 is going to have a C value of 2, and a cell that has begun the process of meiosis or mitosis, but the chromosomes have not been pulled apart yet and the chromatids have not been pulled apart yet, is going to have a C value of 4. A special thing that occurs during meiosis that does not occur during mitosis is crossing over, or recombination it's also called. Prophase in meiosis, prophase 1, is quite a bit longer than prophase in mitosis. And in fact, prophase 1 has its own set of stages, but we're not going to get into that. But crossing over can occur between non-sister chromatids. So here at the left, we've got a tetrad. We've got 2 sets of information. The colors aren't quite right on the legend there for some reason. Blue should be from your dad. But this tetrad is going to consist of 2 homologous chromosomes. Each of those chromosomes is made up of sister chromatids. Non-sister chromatids can exchange bits and pieces, and that's what you're seeing in this recombination event. They stay kind of tangled, so this crossing over persists until meiosis 1. And then after that, the chromatids will be pulled apart. It doesn't always go as planned, and that can create issues. That can cause a cell beginning into meiosis to abort, or it could cause situations where the tetrads don't separate properly and we end up with aneuploidy. So here's an example of crossing over. In that photo there, you're seeing what it actually looks like under an electron microscope. A chiasma is the site of crossing over, and as you can see, we have bits of information that are being exchanged. So remember, meiosis 1 is going to separate homologous chromosomes. Meiosis 2 is going to separate chromatids. So I want to go back and look at a slide that you have earlier in your notes, but this time apply what we know about ploidy and C values. Let's look at this condition again. We've got a 2n equals 6 cell that's going to divide by mitosis or meiosis. Now remember that after chromosome replication, so after the S phase, we have doubled the amount of DNA, but we have not changed the number of chromosomes, and we have not changed the ploidy. So looking at mitosis first, the condition for this cell here, and you'll have to excuse my writing, is still 2n equals 6. We still have 6 chromosomes, except each of the chromosomes consists of 2 chromatids now. Now the C value is higher than it was. It's doubled. We started with a C value of 2. And that's because we have twice the amount of DNA that we would see in the lowest value, which is found in the gamete. And we'll come back to that. So the original cell, 2n equals 6, C equals 2. The next cell here, after going through the S phase, is 2n equals 6 still, but it's C equals 4. Okay. Now these chromosomes are going to line up along the metaphase plate, and nothing's going to change. We still have 2 sets of information contained in 6 chromosomes, and our C value is still 4. And then the final stage here, we've got our cells splitting off from each other, and the chromatids have been pulled to opposite poles before that happened, of course. We've still got a value of 2n equals 6, but now our C value is back to 2. All right, now let's look at what happens in meiosis, because it's probably a bit more confusing. In prophase 1, what happens is the homologous chromosomes come together and pair up and form tetrads. So what happened is we had a stage just like you see in the first diagram of mitosis, but immediately this chromosome and this chromosome paired up, and this chromosome and this chromosome paired up, and this chromosome and this chromosome paired up, and they attach to each other. Now, what we've got, we still have a condition of 2n equals 6, and it's important to note that we are counting chromosomes. We aren't counting tetrads. So we still have a value of 2n equals 6, and we have a value of c equals 4. At metaphase, still the same. Nothing has actually changed at this point. So 2n equals 6, c equals 4. But now, after our first division of meiosis, notice that we only have 3 chromosomes now. So this is a change. This is a noticeable change, and our value is n equals 3. N meaning it's haploid, 3 meaning we have 3 chromosomes. And also we've cut down the amount of DNA. So we've cut down the C value. Probably a useful way to think of the C value is the number of pieces, individual pieces of DNA, or perhaps the weight of the DNA, or the number of base pairs. So the C is going to be 2, our C value is 2. And then in the final state here, after the second division of meiosis, N is still going to be 3. So N equals 3 for all of these cells still. But now the C value has been decreased again because the chromatids have been pulled apart. So this is C equals 1. So the C value is assigned a value of 1 in the cells within the organism we're talking about that have the lowest amount of DNA. So you don't have any cells that have less DNA than your gametes. That's assigned a value of 1. If we have a cell that has twice as much DNA, so let's say we weighed the DNA and we find that there's twice as much DNA, that would be a C value of 2. If we have a cell that has twice that amount of DNA, we have a C value of 4. And those are the only 3 values you can have in human cells. Let's quickly overview the process of meiosis step by step. So again, we have the 2 divisions, meiosis 1 and meiosis 2. The first prophase, the prophase of meiosis 1, is quite a bit longer than the second prophase, and this is where a whole lot of unusual things occur, things that don't happen in mitosis. So in prophase 1, we have the homologous chromosomes coming together and forming tetrads, and we have these chiasma forming. And the chiasma are where the chromosomes will swap bits and pieces of information. So that's the site of crossing over, also called recombination. Towards the end of prophase, we have the breakdown of the nuclear envelope, and then the chromosomes, as tetrads, are going to move to the metaphase plate. So as metaphase begins, the chromosomes are still attached to each other. You can see these black areas in the middle of the chromosome. Those are meant to represent the kinetochores. The kinetochores contain motor proteins that will attach to the microtubules of the meiotic spindle, and that will allow the chromosomes to walk along those railway tracks towards the poles. Anaphase, we've got the chromosomes separated, and you can see that crossing over or recombination has occurred. All the chromosomes are a little bit different now. The sister chromatids are still attached to each other. Then telophase is where the chromosomes get to the opposite poles. We're going to have the reformation of the nuclear envelope, and then cytokinesis is going to divide this cell into 2 cells. Now we start into our second division. So we've got 2 cells that are in prophase 2. The nuclear envelope is breaking down again. In fact, in many animals, the nuclear envelope doesn't bother forming between meiosis 1 and 2, but we'll ignore that. The nuclear envelope is going to break down if it's present, and we're going to end up with our homologous chromosomes lining up along the metaphase plate. The second division looks a lot like mitosis. It basically is the same as mitosis, except that we're starting off with half the number of chromosomes and half the amount of DNA. So these chromosomes line up on the metaphase plate, and just like before, the kinetochore is going to attach to the appropriate microtubule, hopefully, and that's going to walk the chromosomes along the microtubule towards the poles. Then we have anaphase, just like in mitosis, and telophase and cytokinesis, just like in mitosis. And we end up with 4 genetically distinct daughter cells. Now, it is important to note that this process doesn't generate new information. So if you're producing gametes, your gametes contain information that was found in the diploid cell that started the process, but the information has been rearranged so that you will have genes from your mother and genes from your father now on the same chromosomes, and that wasn't the case before. Before, the 2 sets of information were entirely separate. The timing of meiosis is quite different in males and females, in humans and other mammals. Let's look at males first because that's a bit simpler. We start off with this population of cells that are known as germline cells. So there are cells that are set aside in the very early embryo that will become the cells that can potentially undergo meiosis. And these cells, of course, are going to be maintained within the testes. The cells can increase in number during development and during childhood by mitosis. After puberty, some of these cells will also start undergoing meiosis. So some of these cells will become spermatocytes and then divide through the 2 divisions of meiosis to produce 4 haploid sperm cells. This population is maintained by mitosis throughout adulthood. So after puberty, males start making sperm, and they're making sperm fresh all the time, and this continues into old age. Now, what happens in females is a bit different, and admittedly it's not fair, but this is the way the biology works. In females, we start off with that same population of germline cells. Before your birth, those cells enter into meiosis. They start into meiosis and they stop in prophase 1, so they don't get very far. And they're arrested. They go into dormancy at that state. These are cells that could ultimately give rise to egg cells, and all the cells that you will ever have that can do that are present before you're born. These cells are maintained through childhood, but they don't divide or anything. They're in this arrested state. Then at puberty, each month, one of these cells will continue and complete the first meiotic division. That cell will be released into the fallopian tube, also called the oviduct, and it will flow down that tube. If it doesn't meet a sperm cell, it's just going to be shed from your body. It's never going to go into the second division of meiosis. But if it meets up with a sperm cell, the binding of the sperm cell to the membrane of this cell will cause it to enter into meiosis II. It'll finish meiosis II, and after that has happened, then the haploid nucleus of the sperm cell and the haploid nucleus of the egg cell can fuse, and then we get a zygote. Keep in mind that meiosis I and meiosis II will produce polar cells, polar nuclei, that aren't going to be involved any further. Meiosis doesn't always go as planned. The most common mishap that can occur is something called non-disjunction. So this is where the homologous chromosomes fail to separate, or the sister chromatids fail to separate. This is far more likely to occur in females, and the reason for that is that males are constantly producing brand new spermatocytes and brand new sperm, and that population is being refreshed constantly. Whereas the population of cells that produce eggs in females, they've been around a long time. Those cells have been arrested in prophase 1. And there's lots of stuff that can go wrong during that period. So those cells can become damaged and can be unable to repair that damage. And this can cause some issues with the attachment of the chromosomes to each other or their attachment to the mitotic spindle. There are two stages where non-disjunction might occur. It could occur during metaphase 1 or metaphase 2. So the upper diagram on the left shows non-disjunction during metaphase 1 of meiosis 1. So you've got this hypothetical cell that has 2 chromosomes. We've got 2 tetrads that formed, and you can see the top one is separating normally, but the bottom one is not. The homologous chromosomes are not pulling apart. They're staying together. There could be a few causes for this. It could be that the proteins that hold the homologous chromosomes together are not being broken down. It could be that the points of crossing over are not being resolved, so they're remaining kind of tangled up. Or it could be that all 4 chromatids have attached themselves to the same microtubule, and they're all being pulled by their kinetochores to the same pole. Whatever the reason, we're going to end up after meiosis 1 with one cell that has an extra chromosome and one that is missing a chromosome. And after the second division, which, assuming it goes normally, we're going to have 2 cells, so half of our cells, that have an extra chromosome and 2 that have a chromosome missing. Now let's look at the second case here. So on the right, we've got non-disjunction occurring during meiosis 2. Same idea, except in this case, the first division went properly, so the chromosomes separated properly, but the chromatids did not separate properly during the second division. And note that this is going to result in 2 cells that are normal, so they're just N, and then we're going to have 2 cells that are unusual in that they have an unusual number of chromosomes, and that condition is known as aneuploidy. So we have one cell that has an extra one and one that is missing one. Now imagine that we have an egg cell that has an extra chromosome, and that's what's being shown down the bottom here. And it fuses with a normal sperm cell. What we're going to end up with is a zygote that has an extra chromosome. So we would say it has the condition of 2n plus 1. Trisomy 21 is probably the best known example of aneuploidy. And again, aneuploidy is where you have an unusual number of chromosomes. Typically, if you're missing a chromosome, that is a very bad thing. And usually the zygote or the embryo will abort. You won't get development to term. With trisomy 21, you've got extra information. So you've got an extra copy of chromosome 21. And you might think, well, what's the problem with having extra DNA? The problem is that, let's assume all of those chromosomes are turned on and functional. You're going to be producing more of the protein that's coded for by the genes on that chromosome than you should be. You're going to be producing a third more product from chromosome 21. And that can cause you to have more of a certain enzyme. That can cause developmental issues. So with trisomy 21, people that have this, they live full lives typically, but of course there are a lot of cognitive issues and other issues. I want to do a bit of genetics, not very much. It's going to be kind of introductory level stuff, but I would feel remiss if I didn't go over this with you. Let's take a look at a simple biochemical pathway. So remember, a biochemical pathway is where you have a precursor. This is our precursor up here. It's actually a derivative from an amino acid, and then that's going to be converted into something else by a chemical reaction. So the next step is this intermediate here. And there is an enzyme that catalyzes that step. So there's going to be several steps in this pathway, and in fact this is greatly simplified. There's lots more steps than this, but we're interested in an enzyme that's essential for this pathway to work, and that's this enzyme here, tyrosinase. Notice that it catalyzes 3 steps in this pathway. It's an important enzyme. If you don't have that enzyme, then you can't make these pigments. So notice that we have a branch of this whole metabolic pathway that produces eumelanin. That's the black pigment. We have another branch that produces pheomelanin, which is the red pigment. If you don't have functional tyrosinase, the reactions that are required to make these pigments can't occur, and you can't produce any pigment. You have something called albinism. Now let's imagine that you have received two functional forms of the gene that codes for tyrosinase. Then you're going to be able to complete this pathway, and you're going to be able to produce pigments. Let's say you have only one functional copy. So you've got one mutated copy from one of your parents. It doesn't work, and you've got a fully functional form of the gene from another parent. That means in every one of your cells, you're going to have one functional form of the enzyme, one functional form of the gene that codes for the enzyme, and that's plenty. Enzymes, remember, are used over and over again. Typically, you don't need a whole lot of an enzyme. So having two functional forms of the gene for the enzyme is fine. Having just one is fine. But if both genes that you got from both parents don't work, if they're both mutated, then you can't produce any tyrosinase and you can't produce pigment. For the most common form of a gene in a population, we symbolize that using uppercase letters. Typically, this is the fully functional form of a gene. For what's called the null version, that's the version that doesn't work, we use a lowercase letter. So let's say we have a person that has this genotype, little a. That means they have two non-functional forms of the gene. And let's say we have another person that has this genotype. That means they have two fully functional forms of the gene. Let's just say for the sake of argument that this individual up here is a male. And that means that they are going to produce sperm, and the sperm will all have one form of the gene because the sperm are haploid. And the only type of sperm they can produce are sperm that have this functional form. Now let's say, again, just for the sake of argument, that this is a female down here, and hopefully you can make out what my symbols mean there. The female can produce eggs that have the non-functional form, or she can produce eggs that have the non-functional form. That's all she can produce because she doesn't have a functional form. But the eggs are going to be haploid, and what you're seeing here, this and this, those are the genotypes of the sperm. And what you're seeing here, these are the genotypes of the eggs. And we can do something called a Punnett square analysis to figure out the possible outcomes. So what we're looking for are all the different outcomes for the mating of these two individuals. And it turns out there's only one outcome. So the father can only donate a dominant form, that's the functional form of the gene, and the mother can only donate the recessive form, that's the non-functional form of the gene. And we're going to have these four different outcomes, which are all the same, and this is the heterozygote condition. Heterozygous refers to the fact that in the zygote, we're going to have two different forms of that gene. So again, the big A is dominant. That means that it overrides the effect of the recessive gene. A homozygous individual is going to have two forms of the gene that are the same. So our parents here, this is a homozygous individual, homozygous genotype. This is a homozygous genotype because they both have two alleles, two forms of the gene that are the same, and their offspring are heterozygous in this case. Alternate versions of a gene are known as alleles. And we can have several different versions, but in the simplest case, and the case we're going to look at, we have two forms. We have one that's fully functional, and we have one that's non-functional. Now, like I said, we can have other forms. We can have genes that code for receptors that work, but they don't work very well, or that code for enzymes that partially work, or they do something a bit different. But again, in the most simplest case, we're talking about genes that are either functional or not functional. The functional form in this case would be the dominant form. So looking at albinism, if you have two recessive alleles for that tyrosinase gene, you're going to see albinism occurring. But if you have at least one of the fully functional genes, that will override or be dominant to the recessive condition. You've probably heard of Mendel before. I'm not going to get into him, but I invite you to read more on this man. So I want to work through a couple examples really quickly, and bear with me because I'm trying to do this with a mouse, and it's going to look like a little child wrote it. But anyway, let's start with complete dominance. This is where one allele is dominant over another, and that's what we were talking about with albinism. So let's continue with that example. Let's say we have an individual that is heterozygous. That means they have this condition, they have one dominant form of the gene and one recessive. And let's say that they have a child that has albinism, and they have two non-functional forms of that gene. Now, if we want to figure out the probability of outcomes for this mating, and a mating is shown just using an X, by the way, we can use a Punnett square to do that. And let's just say that our first individual, we're going to put them on the top here, they can produce gametes that are either that or that. And again, it's really important, students always get this mixed up. On the Punnett square, what you are doing is you are crossing gametes. And this is the genotype for that particular locus or location on the chromosome for that particular gene for the sperm or egg. So from this individual, they can only produce gametes that have one copy of the gene, and that copy in half of the gametes will be the dominant form, and in half of the copies of gametes produced, it will be the recessive form. So the other individual can produce gametes that have either that or that. They're all going to be the same. Now the outcome is going to be what happens when the gametes come together during fertilization to produce a zygote. So we could have this as an outcome. We're just adding those two things together. That would be known as a heterozygous condition, or heterozygote condition, sorry. So in a heterozygous individual, in the zygote, we're going to have two different forms of the gene. That's what the hetero part means. So this, of course, would also be the same. And then if we combine these two, we're going to get what's called the homozygous recessive condition. Homo meaning the same, so in the zygote, we have only one version of the gene present. So these individuals here are going to have the recessive condition, which is albinism. The other individuals here are going to be carriers for this condition because they're carrying that recessive allele, but they are going to have normal functioning tyrosinase, at least enough of it to do the job, and they will be able to produce pigment. Codominance is where we're looking at two alleles, and one allele does not override the effects of the other. Instead, we see a combined outcome. We see the effects of each. And a good example of this would be blood typing, so ABO blood typing. Now, with ABO blood typing, we're looking at a specific location on a protein, and we're looking at whether or not certain sugars are present. And we can have three conditions. We can have an allele of this gene where there is no sugar present at that location. This is a recessive allele, and I'm going to show it as a lowercase I. We can have an allele that codes for the A form of that antigen. And we can have another allele that codes for the B form. Now there are other ways to write these symbols, but this is the way I like to do it. You can use another way if I give you a question and you have to solve it, but this is the way that I prefer. Now note that the A form is dominant to the recessive form. So this here is dominant to this. It overrides the effect of that. However, this A form does not override the B, and the B does not override the A. The B does override the recessive form, the lowercase I. So we have a case here of both codominance between the A and B form, and that's why they're uppercase I's with the superscript A or B. But these dominant alleles are not dominant to each other. So I'll tell you what I mean here. So let's say we have an individual that is blood type A. There's a few ways we could arrive at that. So we could have an individual that has 2 functional copies, because remember, we're talking about diploid cells here. So that could be the genotype for an individual that has blood type A. Or they could be like this. They could have 1 functional form, and it's the A form, and 1 non-functional form. So if that's the case, they still produce enough antigen of the A type to be recognized as being A type blood. Now, let's say we have someone who's AB. If they're AB, they have to have this genotype. There's no other way we can have someone who is AB blood type. Okay, so let's take a look at an example here. Let's say we have someone who is A-type blood, and they are of this genotype. And let's say they're going to have a child with someone who is B-type blood, and they are this genotype. Now we'll take a look at the gametes that they could produce. So looking at this first individual here, half of the gametes are going to be like this. Half of the gametes are going to be like that, because again, the gametes are haploid. They can only be one or the other. And the second individual there, half of their gametes are going to be like this, and half of their gametes are going to be like that. And now we just combine these to see the different possible outcomes. So again, this is a probability table basically. So one outcome would be this. So that is an AB individual. One outcome would be this. That would be an A type. One possible outcome would be this. That would be type B. One possible outcome would be this. And this is type O. They aren't expressing any antigens at this location on the red blood cell. All right, and finally, and I'm going to go through this quite quickly, I just want to talk about genetics and family history. And if we're trying to work out family history, we use something called pedigree. And there are some people that work at hospitals that help people sort out the probability that they might have a child with a certain condition, for instance, and they do that by looking at pedigrees. What you're seeing here is a pedigree for the royal family, and you're looking at occurrences of hemophilia in a family tree. So first of all, something to point out, when you have a square, that represents a male. When you have a circle, that represents a female. If the circle or square is completely filled in, that tells you that the person has the condition we're interested in. So in this situation, the squares that are filled in, those are men that had hemophilia. In this particular case, they've used a partially filled in shape to indicate that the person is a carrier. So Victoria there, for instance, was a carrier for hemophilia. In fact, it's suspected that the gene mutated in her that's responsible for hemophilia. Hemophilia is where your blood doesn't clot correctly. And then she passed this down to her offspring, and they passed it down to their offspring. Wherever you have a square and a circle connected by a line, that means that they've mated, and then whatever is coming off of that line is their offspring, their children. So yeah, this is the pedigree for the royal family, again, starting with Queen Victoria, probably the most powerful person to have ever lived on the planet. So the symbols here. Again, a square is a male, a circle is a female, a line between them, that's a mating, and then if we've got lines coming off of that leading to other squares and circles, those are the outcomes of that mating. So we have a boy and a girl here being born to mother and father. There's some other symbols here that aren't used as commonly, so dizygotic twins, monozygotic twins, etc. I won't get into those, they're not used all that often. The affected individuals are going to be shaded in, as you can see, and sometimes, but not always, people that are heterozygous or carriers, the symbol is filled in partially, but they don't always do that on a pedigree. The proband, that's the individual that you're doing the analysis for. So let's say that you had someone come in and they were quite concerned that if they had a child, it might have a certain disease, and you want to work out the probability. You would draw out their family tree as a pedigree, you'd put an arrow next to them. That indicates that this is the person you're trying to work out the situation for. Another thing down the bottom there, note that we've got consanguineous marriages. These are marriages between cousins. That creates problems. There's a reason that that's taboo and not legally allowed. The reason for that is if you have a recessive allele that runs in your family, it's more likely to pop up in children of people who are closely related. So let's look at an example of a few different pedigrees. The first one is for people who have some sort of deficit with their mitochondria. And that deficit is a result of one of the mitochondrial genes being broken. So in the mitochondria, there is a circle of DNA. This is part of the excellent evidence that suggests mitochondria are derived from bacteria. That circle of DNA doesn't contain very much information, but it's really important information. So there's 37 genes there. If one of them is damaged, usually that's really quite bad. At the very least, you're going to be quite lethargic, and that's because you can't make as much ATP as you should. And that's going to probably affect development as well. But what's interesting about mitochondria is they're passed down only from the mother. At least that's the case in humans and most mammals. The reason is that cytoplasm is something that's donated by the mother but not by the father during fertilization. So the sperm has almost no cytoplasm. It's just a nucleus, and then of course you've got a tail attached to the plasma membrane that surrounds that nucleus, but there's very little cytoplasm. And also the mitochondria in the sperm are attached directly to the tail. There's lots of mitochondria in sperm because they're very energetic, but they stay attached to the tail and they don't enter into the egg during fertilization. Only the nucleus goes into the egg. The egg cell is at least 100 times bigger than the sperm cell, and it contains a lot of cytoplasm and a lot of nutrients and a lot of things that are needed to get development going in the zygote. So that initial cytoplasm in the zygote came from the mother's egg. And the mother's egg, of course, contained mitochondria. And the mitochondria in the zygote, therefore, all came from the mother. So if there is an issue with a mitochondrial gene, if that issue is in a father, it won't be passed to any of his children. If it's in a mother, it'll be passed to all of her children, male and female. So notice it's inherited only through the mother, and all of the offspring are going to be affected if the mother is affected. Autosomal dominant inheritance is where a certain trait or disorder is caused by the presence of a dominant gene. That's not terribly common. Usually disorders are the result of recessive genes, but there are a few conditions that work this way. For instance, polydactyly, which is where you have too many fingers or toes. So in this case, if a person has the dominant allele, they will show the condition, and the people that are showing the condition here, their shapes have been filled in. Conditions that are autosomal recessive require 2 recessive alleles in order for us to see the recessive phenotype. So an example of this that we've talked about a few times now would be albinism. We only see albinism in people that have 2 recessive alleles, so they're homozygous recessive. So here's a family tree or pedigree. You can see that we've got the generations numbered with Roman numerals, and then each of the individuals is numbered within each generation. So looking at the top here, generation 1, we've got 1 and 2, so male and female, and we don't know their genotype for sure. We know that they have a dominant allele because they're not showing the homozygous recessive phenotype or condition. But we don't know what the second allele is. It could be another dominant allele or it could be a recessive. And normally when you're doing these sorts of analyses, you aren't sequencing DNA. Instead, you're looking at the person's phenotype, which means, you know, what you can see or test for without testing the DNA. Now notice in generation 3, between individuals 5 and 6, we have a consanguineous mating. Consanguineous means same blood. So we're dealing with 2 close blood relatives here, 2 cousins. The reason that this kind of mating is discouraged is because if you have recessive traits that are harmful running in your family, they're far more likely to pop up as a homozygous recessive state if you have matings between closely related individuals. So it doesn't mean if you have a child with your cousin that your cousin's going to have 2 heads or be mutated. They might be totally fine. And in fact, Charles Darwin had lots of children with his first cousin, and they turned out pretty well. But if you do have something that runs in the family, like I said, it's far more likely to cause problems in your children because it's far more likely to pop up as a homozygous recessive condition. Most disorders are caused by recessive alleles. So here's an example here. You can't always interpret them for sure, and I'll let you go through this and try it yourself if you want. But this is most likely an autosomal dominant situation because the trait pops up every single generation. Autosomal recessive traits are rarer. They don't pop up as often. So I have 2 more pedigrees to look at, and these both have to do with X-linked traits. Those are traits that are coded for by genes on the X chromosome. Remember that the Y chromosome has the SRY gene. That's important for determining sex, but there's really not a whole lot more going on the Y chromosome. On the X chromosome, on the other hand, there's about 1,000 genes. And those genes can be dominant or recessive, just like the genes on the autosomes. And we depict them in the same way. So we're going to use a lowercase letter for the recessive form and an uppercase letter for the dominant form. So looking at our pedigree here, generation 1, individual number 1, is a female. And that means she's got 2 X chromosomes. And this particular female has 2 copies of the dominant allele for the gene that we're interested in. Number 2 is a male, and remember the male is going to be XY, so that's the way you have to write it. There's only 1 X chromosome, and this particular male has a recessive form of that X. And that's going to produce some sort of condition or maybe a disorder, maybe color blindness, for instance, something like that. So this individual is showing that phenotype, they're showing that trait. Now, when they have children, the male child, number 1 in generation 2, got the Y from his father. So a boy cannot get an X from his father because he had to get the Y from his father. And the sperm is either going to have an X or a Y, not both. So the X had to have come from the mother. So we can see that the X has the dominant allele because that's the only type of allele that the mother has. That's the only thing that the son could have gotten. Now we have a daughter, number 2 in generation 2, that is heterozygous. So they had to get an X from their father, and they got an X from their mother. So again, for any child of a mating, they will have to get an X from the mother, and then they could get another X from the father, or they could get a Y from a father. Okay, this individual is going to mate with individual 3, so that's an individual from outside the family, and this is a male that has the dominant form. Now note that in the 3rd generation, child number 1 is showing the trait, and that's because they have a recessive allele that they got from their mother. The other children are not showing the trait. So number 2, it's a male, but he got the dominant form. And then the 2 females that we see there, they have at least 1 copy of the dominant form. Now the big thing that needs to be pointed out for X-linked recessive traits is that they pop up more commonly in males. And that's because if a male gets the recessive allele, he doesn't have another X to counteract it. So he will show the trait. And back to hemophilia. Hemophilia is the result of a gene on the X chromosome. In fact, there's a few genes that can be involved. But these are genes that are essential for proper blood clotting. And this is the whole family tree that I talked about briefly before. So we're starting with Victoria here. Again, Victoria is thought to have been where this particular mutation originated for this particular tree. And that's because it seems pretty likely that neither of her parents had this condition. So you can see it's popping up in a lot of males. So the only place that we see the actual trait of hemophilia in the royal family is in males. There are quite a few females that were carriers, so they had 1 recessive form on one of their X chromosomes, but it was balanced by a dominant form. And those individuals are shown as the half shaded circles. And finally, we can have X-linked dominant. So this is where the dominant form of a gene on the X chromosome is responsible for the trait that we see. And you can see here that if the father has this condition, all of the daughters have to have the condition because they have to get 1 X from their father. And that 1 X will override the effects of the other X they get from their mother if it's a recessive. So it's something that pops up more commonly. And again, the main thing that will help you identify this is that a father with the condition will pass it to all of his female children.